All right, so chapter 9, it's a fairly long chapter. That's why I pair it with 8, which is a short chapter. And so what I want to do, we're not going to have time to cover everything in detail. So what I want to do is go over the PowerPoints and point out the most important aspect and go over the most important aspect of each of the PowerPoints. There are recordings on eCampus of all of these chapters in which you go through I go through every one of the PowerPoints. But I thought since we don't have time, we have about an hour and a half, and that's not enough to go through the entire all 3 PowerPoints. At least we can go over what's most important. So I thought that's how we're going to approach it. And obviously, if you have questions at any moment, just go ahead and interrupt, and we can address the questions. So part of what this chapter is going to do is going to go back to chapter 4, tissues, and go over muscle tissues again. The last part of this chapter does that. It goes back to muscle tissues and compare and contrast the skeletal muscle, cardiac muscle, and smooth muscle. However, most of the chapter is focusing on skeletal muscle. So that's the first thing, is go back to muscles and refresh yourself on the characteristics of muscle tissue, and then more specifically skeletal, cardiac, and smooth muscle. So that's the first thing that you would need to do. So I'm not going to go through these because this is, the stuff that we've already covered. Go over the functions of the skeletal muscle, and we covered these in chapter 1. And nothing here is really that surprising. We talked about the role of the skeletal muscle in body temperature on chapter 1. This is something was not in chapter 1, but still, you kind of have an idea that entrances and exits in the body are surrounded by muscle. So there are going to be circular muscles around these openings, like mouth, eyes, anus. Okay, so none of this is very surprising, actually. This right here, the organization of the skeletal muscle. This is actually one of the challenges of the chapter, is to keep in mind or understand how skeletal muscle is organized from its gross anatomy all the way to the microscopic anatomy. And this information that we have here in this PowerPoint takes a bit of both, gross and microscopic. Most of it is microscopic, however. So essentially, the best way to approach this is with a picture. This is going to take us from the gross anatomy, from the muscle, all the way to the muscle cell. And here's one of the challenges with this material, is that muscle cells are actually called muscle fibers. So that's the first thing to remember. Every time you see muscle fiber, that actually means a muscle cell. It's the same thing. Another name for muscle cell or muscle fiber is going to be myocyte. My means muscle. And then you just put the combining vowel, which is O, and then cyte for cell. So a myocyte is a muscle cell. So any of these terms will refer to muscle cells. You should know that, and when you encounter any of these cells, you should know that they're referring to the muscle cell. Okay. So we're going to go again all the way from the entire muscle to the individual cell, and we're going to look at how each of these structures are organized and the connective tissue that surrounds these structures. Now, at this point, it should be no surprise that there will be connective tissue surrounding muscles. We found connective tissue surrounding organs, like the pleura in the lungs, the pericardium in the heart, peritoneum in the organs of the abdominal cavity. So that's no surprise that muscles, periosteum on bones. So there should be no surprise that muscles also have their connective tissue. So the connective tissue that surrounds an entire muscle is the epimysium. So next time, if you're preparing a roast or something like that, a big piece of meat, notice the translucent membrane that sometimes covers the outside of the piece of meat. That's the epimysium. Okay, so then you take the muscle and you look at how it's organized. And the way that if you were to cut it a cross section, look at it across, this would be this yellow thing here would be the epimysium. You see these circular structures here. You would need a dissection scope to see these. And these circular structures are made of connective tissue that surround muscle cells. So each one of these circles is called a fascicle right here. So this is one of the circles right there. The smaller circles inside the circle are the muscle cells, and the connective tissue that brings these fascicles together, or these muscle cells together, is called the perimysium. So epimysium was first. Second is the perimysium. Epimysium surrounds the entire muscle. The perimysium surrounds a group of muscle cells, which we call a fascicle. Then when you take out one of these muscle cells and you look at it individually, right here, the muscle cell has a connective tissue surrounding it, which will be outside of the cell membrane. And that connective tissue membrane is called the endomysium. So that's number 3, the endomysium. So epimysium, perimysium, and endomysium. Okay, so then we're going to go further than this here in just a second. For now, this is a microscope picture. I was trying to look for the magnification. I don't see it anywhere. But this is a muscle of a microscope picture of a cross section of a muscle. And so these right here will be a fascicle, which means that the white tissue surrounding it is the perimysium, and the smaller red circles inside are going to be the muscle cells. The white area around the red circle will be the endomysium. And then if you focus on one of the or several of these cells inside the fascicle, you'll see the actual cell, and surrounding it, the endomysium. So again, the endomysium is outside of the cell membrane. All right, so then we're going to focus on the muscle cell itself. And a skeletal muscle cell is a very specialized cell. It doesn't look like any quote-unquote normal cell that we might have to think about or seen in biology 1406, for example. These are specialized cells that can actually create or receive an impulse from a nervous system neuron and create an impulse itself that would cause a contraction. Okay, so because of the specialized function, you would expect specialized structures, and that's exactly what you're going to see. First of all, the cell membrane of the muscle cell, the skeletal muscle cell, is not going to be called a cell membrane. It's going to be called sarcolemma. So you should know the sarcolemma is the cell membrane. The cytoplasm of the muscle cell is called the sarcoplasma. Sarco is ARCO, means flesh. Okay, so the endoplasmic reticulum of the muscle cell is called sarcoplasmic reticulum, or SR. Remember that endoplasmic reticulum was made of membranes, and there was a smooth endoplasmic reticulum and rough endoplasmic reticulum. The acronym was ER, and so there was a smooth ER and rough ER. Rough ER was involved in protein synthesis. The smooth ER was the storage place. Does the sarcolemma touch the endomysium? Yes, it is immediately deep to the endomysium. Yeah, it does. So the two of them will be in contact. Yeah. And the endomysium is connective tissue, so there will be fibers in there. And so there may be proteins from the cell membrane that reach all the way up to the endomysium. So because the sarcoplasmic reticulum is an endoplasmic reticulum, it's actually a smooth endoplasmic reticulum, to be more specific. There we go. It's a smooth endoplasmic reticulum. So this is not the endoplasmic reticulum involved in protein synthesis. This is an endoplasmic reticulum that is there for a storage, modification of molecules. In the case of the skeletal muscle fiber, is storage of ions more specifically calcium. We're going to see these here in a bit. I'm just kind of getting ahead of myself here. Okay. So know these three terms, definitely, and then we'll talk about the other ones here in just a second. So first thing about the skeletal muscle cell, if you remember from chapter 4, it was a huge cell that was multinucleated. Anytime you have a multinucleated cell, that means that the cell was made during development by the fusion of many cells, and that's why you end up with a multinucleated cell, and that's why this is such a big cell. So here's the sarcolemma, which is the cell membrane right there. Okay, the cell inside is loaded with these contractile proteins called myofibrils, and that's what you can see here. These rod-looking things are the myofibrils, and again, this is something unique. You don't see these in other cells. And there are so many of these myofibrils packaged inside the skeletal muscle cell that there is hardly any room for the other organelles. So the other organelles, like the nuclei, have to be pushed to the periphery of the cell. Okay, so we have these myofibrils, which are going to be contractile proteins. Surrounding the myofibrils, these blue things right here, is going to be the sarcoplasmic reticulum, the smooth endoplasmic reticulum. This is the sarcoplasmic reticulum of the cell. These sac-looking structures right there that surround the myofibril. And when you see something like that that is so unique, you should suspect that this arrangement has something to do with function. Notice the yellow tube is spaced in between these sarcoplasmic reticulums. These yellow tubes are called T-tubules. Oh yeah, there we go, T-tubules, okay? And they're made by invaginations, infoldings of the cell membrane. So the cell membrane folds inward and creates these tubes that we call the T-tubules. So the T-tubules. And notice that on either side, the T-tubules are flanked by sarcoplasmic reticulum. So the sarcoplasmic reticulum here, sarcoplasmic reticulum over here, and in the middle, the T-tubule. This arrangement is unique to skeletal muscle. It is called a triad. So this right here is a triad. And it is not found in any other type of muscle. Cardiac muscle doesn't have it. Smooth muscle does not have it. So this is unique to skeletal muscle, which tells you this has something to do with the unique physiology of the muscle. The area of the sarcoplasmic reticulum that flanks the T-tubules on either side are called the cisternae, terminal cisternae. So we essentially have one terminal cisternae, a T-tubule, and another terminal cisternae. Notice that the T-tubule is essentially a connection to the outside. So something from the outside is going to travel inward following the T-tubule. And whatever that something is going to have an effect on the sarcoplasmic reticulum. All right, so that's an important feature, a unique feature of a skeletal muscle cells. We definitely should be familiar with it. This is a close-up of a triad. Again, this is the sarcoplasmic reticulum, and it is filled with calcium ions. Another sarcoplasmic reticulum on the other side, filled with calcium ions, the T-tubule in the middle. That's a triad. So you should know what a triad is. And this is showing you the relationship between the T-tubule and the triad. And essentially what's going to happen will be that the nervous impulse, which is commanding the cell to contract, is coming from the neuron, and the impulse will be transferred from the neuron to the muscle cell. It will travel inside the muscle cell via the T-tubule, and that signal will open calcium channels. on the cisterna, which are connected to the T-tubules, and calcium will leave its storage place and begin to fill the inside, the cytoplasm of the cell, or sarcoplasm of the cell, okay, and initiate muscle contraction. So that tells you immediately calcium has something to do with muscle contraction. And the calcium is coming from inside the cell, from the storage place, which is the cisterna or cisternae of the sarcoplasmic reticulum. So that's a little hint to the physiology of muscle. Now, one last thing to mention in skeletal muscle structures. The myofibril, I said, was a contractile protein. So the myofibril is going to contract, and as the myofibrils contract, the entire muscle cell is going to contract. So the next question would be, what is it in the myofibril that contracts? In other words, what is the myofibril made of? The myofibril is made of a repeated row or arrangement of contractile proteins, which is called a sarcomere, which is this right here would be a sarcomere right there. Let me give you a better view of the sarcomere. Here's a sarcomere. From here to here, this arrangement of contractile proteins, which you can see here in more detail, that's a sarcomere. Now, when it comes to muscle contraction, that is as far as you can go. That is the unit of muscle contraction, the sarcomere. When the sarcomere contracts, and it's going to contract towards the middle, the entire myofibril contracts. The myofibrils are made of repeated sarcomeres. When these sarcomeres contract, the myofibril contracts. When the myofibrils contract, the muscle cell contracts, which causes contraction of the fascicles, which causes contraction of skeletal muscle. So the unit of contraction is the sarcomere. Needless to say, you have to know what a sarcomere is, the unit of muscle contraction. And we're going to go over the structure here in just a second. So if you go back to the organization of muscle, the skeletal muscle, all the way from the muscle, the gross anatomy, to the unit of muscle contraction, the sarcomere, we go from the skeletal muscle to a fascicle. So one of these circles will be the fascicle, surrounded by the perimysium. Then the fascicle is surrounding muscle cells. So you take a single muscle cell surrounded by an endomysium, which is on the outside of the sarcolemma, the cell membrane, and the muscle cell is loaded with these little myofibril contractile rods. You take one of those out, and it is made of repeated sarcomeres. They're enveloped by these triads also. You take one sarcomere right there, and that's as far as you can go. There's the unit of contraction, the sarcomere. So many sarcomeres make one myofibril. Many myofibrils are found in one muscle cell. Many muscle cells are found in one fascicle, many fascicles in one skeletal muscle. So this is like those little Russian dolls that you keep opening the one on the outside, and there's a smaller one on the inside. Well, this is exactly the same thing. You get the smaller and smaller and smaller component until you get to the unit, which is the sarcomere. So definitely know that. This is basic information. So you should expect questions about these structures. And definitely, I cannot say definitely enough times, know the structure of the sarcomere. When it comes to the test, the structures that I have available to put on the test will be the structures that you see in the worksheets, structures in the PowerPoints, but especially the ones in the worksheet, because those are the ones I made myself. So I have access to those pictures. Access to these pictures in the PowerPoint sometimes is not as easy. So this is to say, when it comes time to understanding the structure of a sarcomere, expect to see pictures like the ones in the PowerPoints and in the worksheets. Okay, those are the pictures that should be especially familiar with. Okay, so even though all the pictures same structure, so ultimately you should recognize the same parts, regardless of how the sarcomere is depicted. All right, so sarcomeres, again, a myofibril is made of many sarcomeres. In this case, this is a little myofibril, and you can see that from here to here is one sarcomere. So this is half of another sarcomere, this is half of another sarcomere. When you look at the sarcomere, you can see, let's see, do these, yeah, you can see this red line right there. These red lines going across in the middle of the sarcomere. That will be these red lines right here. And we call those thick filaments. There we go. And then flanking these red lines from either side are thinner lines, which we call thin filaments. So those would be the blue lines that you see there, the thin blue lines right there. Okay. And notice that the thin blue lines do not go all the way across. They kind of stop right there. Then there is a space where there are no thin filaments. And then there's another thin filament on the other side. So there's this area in the middle of the thick filament where there is no thin filament, which you can see right here and right here. That area is called the H zone. So I wouldn't worry about the M line too much. The H zone will be that area right here. That's the H zone. All right, so when scientists began to look at these myofibrils, they were actually huge proteins. I'll come back to these PowerPoints in just a second. They were actually able to take X-ray pictures of it, of the sarcomere, and this is what they would see right there. Okay, so now they had to try to make sense of these pictures, and they began naming the areas of a sarcomere, and they named the beginning of this dark line to the other side of the dark line. So that would be the beginning of the thick filament all the way to the end of the thick filament. They named that area the A band. And so because they were able to see these area that absorb light, that's what A stood for. And they call that the A band. And then they had these areas. There were no thick filaments. There were only thin filaments, and they call that the I band. So they had I band, A band, I band, A band, etc. They didn't understand at the time completely that some of these bands were in the middle of two sarcomeres, but that's where these names come from. The beginning and end of a sarcomere. The sarcomere is flanked by two lines of proteins called the Z lines. Sometimes they're called the Z discs. So Z lines or Z discs. Okay, let me go ahead and clean this out a little bit so we can see them better. All right, so this is one Z line or Z disc. And don't get confused. Sometimes you'll see it called C lines. Other times you'll see them called C discs. Both are correct. All right, there we go. There's one sarcomere. Okay, so notice that the I band is in between two sarcomeres. All right, to me, more important than the A band is the I band, is the A band. Okay, A band. All right, the A band has an area right here where thick and thin filaments overlap. You have thin, thick, thin, thick, thin, thick. This is called the zone of overlap. Then you have the empty or lighter zone, which is the H zone, and then another zone of overlap. So we have two zones of overlap on either side of the H zone. So that's what you can see here. Zone of overlap, H zone, zone of overlap. That's what the A band is made of. I band is essentially one Z disc and two thin filament from two different sarcomeres. Okay. All right. So given a sarcomere, you should be able to identify the A band, the I band, the Z disc, or Z line, the thick filament, the thin filaments, the H zone, all these right here is H zone. Did I mention zone of overlap? I don't remember now if I mentioned zones of overlap. Definitely know the zones of overlap. So this is one zone of overlap. This is another zone of overlap. And just again, by definition, the A band encompasses from the start of the thick filament to the others, for one end to the other end of thick filaments. Now, first of all, this arrangement is what creates the striations that we see in a skeletal muscle. Now, we have striations on a skeletal muscle and we have striations on cardiac muscles. That tells you cardiac and skeletal muscles have sarcomeres. Smooth muscle doesn't have striations. That tells you they don't have sarcomeres. Okay, so that's the first thing. Sarcomeres are going to be found in the skeletal muscle and cardiac muscle, in the striated muscles, because the striations are made by the sarcomeres. Okay, so again, know the bands, know what the A band is made of. Another important point here, and I'm kind of skipping to another PowerPoint because I want to make this point now and then maybe repeat it again a little bit later. Another point is to know what changes, which ones of these structures change when the sarcomere contracts. There's a nice PowerPoint that shows that, which I'm looking for. Okay, just a second. Okay. There we go. All right, there's contraction of a myofibril, which is caused by contraction of the sarcomere. So again, if you have a myofibril and the sarcomeres begin to contract, and by the way, the sarcomeres are going to contract towards the H zone. So from either side of the Z line or Z disc, the sarcomere will contract towards the middle. So each one of the sarcomeres contract towards their middle. Each sarcomere has an H zone. So both of them, every single sarcomere contracts towards their H zone. The entire myofibril contracts. And when myofibrils contract, the entire muscle cell contracts, which causes fascicle contraction, which causes skeletal muscle contraction. All right, now let's take a look at what changes during the contraction. First of all, the A band is not going to change because by definition, the A band is the thick filament from beginning to end, and that's not changing. Okay, A band doesn't change. However, take a look at the H zone. This is the sarcomere at rest. This is the sarcomere when it's contracted. So definitely the H zone is decreasing in size. Zone of overlap. Take a look at the zone of overlap. This is the zone of overlap when the sarcomere is at rest. This is the zone of overlap when the sarcomere is contracting. So the zone of overlap has increased. So the H zone decreases. The zones of overlap increase in size. A band is the same, however. Okay, it's just the parts of the A band have changed in size. Take a look at the I band. Very, very small. Here, the I band went from here all the way to the other side. And now here's the, you can see the purple of the thick filament of the next sarcomere. So the I band is just this area right here. The I band has decreased. What is happening during contraction is that these thick filaments, and you can see that the thick filaments have these little heads going around, are going to grab a hold of the thin filaments and are going to begin to pull the thin filaments towards the middle. And that's what's going to decrease the H zone, because now the zone of overlap increases as the thick filaments pulls the thin filaments. the I band decreases, A band stays the same, that one is not going to change. H zone decreases, zones of overlap increase. So you should definitely know these. What happens to the sarcomere when there's muscle contraction? Now, the other thing you have to know is thick and thin filaments, the proteins that make up thick and thin filaments. Thin filament is the more complex, actually. Thick filament is very simple. Thin filament has 3 proteins. It has a protein called actin, which are these yellow circles here. Each one of these yellow circles is called a G-actin. And notice that these yellow circles assemble themselves into these filament, these long structures. So all of these G-actin proteins assembled together is going to be called an F-actin, F for filament. All right. Notice also that the actin, which is the important protein, has this little black circle here, right there. You can see that. Each one of them has that little mouth open right there. That is called the active site. So we have an active site of actin. Okay. So we have the actin with the active sites, and the active sites are being covered by this brown protein right here called tropomyosin. So we had actin, we have tropomyosin covering the active sites of actin. And then we have this bluish protein here, which is called troponin. And troponin is probably one of the most important proteins in all of these that we're looking at. Because troponin is the regulatory protein. This is the one that does something. So this is what it's going to do. First of all, troponin is grabbing a hold of actin and tropomyosin, kind of holding them together and in place so they don't slip from each other. Troponin, with one of its little free heads, troponin has 3 heads, is going to bind calcium. So this is the calcium binding protein. And calcium is the ion needed for muscle contraction. And this is the reason why calcium is needed. Calcium is going to bind to troponin. When calcium binds to troponin, troponin changes its shape, it moves. When it moves, it drags with it tropomyosin. And tropomyosin now moves out of the way, and the active sites of actin are now exposed, are now free. So again, troponin binds to calcium, troponin moves when bound to calcium, freeing the tropomyosin away from the active sites of actin, and now the active sites of actin are going to be available. They're not hidden anymore. So that's one part of the, that's one of the events, first part of the events that happens during muscle contraction. Now, the other filament, the thick filament, is made by one protein essentially, myosin. And myosin is an elongated protein that has a tail at one end and it has a couple of heads at the other end. And so there's many of these proteins in which all the tails are together, all the heads are together. And the heads are strategically positioned in the same place right around where the actin heads are located. So myosin heads and actin heads. Okay. Let's see if another PowerPoint explains things better. Okay, we got that. Okay, so you can see here the position of the myosin heads close to the thin filament with the actin is. The myosin heads have an actin binding site. So we have actin up here, with an active site. We have myosin heads with an actin binding site. Active site of actin, this is a play on words here, active site, and an actin binding site. The active site of actin is attracted to the actin binding site. So these two sites, one in the actin thin filament, the other one in the head of the myosin, attract each other the same way that two magnets attract each other. And the minute that these two areas are available, they're going to come together. So the only reason why they don't come together during muscle relaxation is because when the muscle is relaxed, the active site of actin is being covered by tropomyosin. And it's not until calcium binds to troponin, and troponin shifts its shape, moves, and drags with it tropomyosin, that the active site of actin is now, oops, I didn't mean to do that. Okay, well, the active site of actin is now free to bind to the head of the myosin. So that's why calcium is needed for muscle contraction. Calcium is going to initiate the events that are going to free the actin active site. The myosin head also has an ATP binding site. Remember from last semester, ATP is the energy currency of the cell. So the molecule that is going to bind energy is going to be the myosin head. And again, this is looking at thin filament and what it's made of. There's the G-actin, which when it combines with other G-actins, will make up the F-actin. This is tropomyosin, and this is troponin, which is the calcium binding. Definitely know all of these proteins. Tropomyosin, troponin, actin, G-actin, F-actin, myosin. Here's the active site of actin being made available, which means calcium has bound to troponin. Definitely know the role of calcium. Proteins that I do not talk about, but they probably are in the textbook, and it's okay. Can you say this sentence again? Calcium initiates. Yeah. Calcium, and I don't know if I can repeat it again verbatim, but essentially calcium initiates muscle contraction because it's calcium that initiates the events that are going to make the actin active site available. They're going to free up the actin active site. So calcium initiates the events that are going to lead to releasing the active site of actin from its hiding place. So that's the role of calcium. And then once that happens, once that active site is released, then the myosin head can bind to it and begin the events of muscle contraction. So, so the proteins I have not mentioned are titin, which is the little protein that holds the thin filaments to the C lines. The other one is, there's titin again. I thought that was, nebulin. Oh, there it is, nebulin, right here, which is another holding. So these titin and nebulin are holding proteins, kind of hold things together. And so I don't mention them. They don't have a physiological role other than just more of an anatomical holding things together. So I kind of ignore them. But if you read in your book and you find them, that's what they are. Okay, so they do exist. I'm ignoring them because I'm concentrating on the ones that have a direct role in muscle physiology. Okay, so I cannot say it enough times. Know the parts of a sarcomere. Let's see. know the proteins that make up the thin filament, know the proteins that make up the thick filament. Be prepared to answer questions like, which of these proteins bind calcium? Okay. Which of these proteins is covering the active site of actin? Here. Which of the proteins can bind to ATP? It's the myosin head. Okay. So when the myosin head and myosin bind, they create what we call a cross bridge. A little cross bridge in here somewhere. Yeah, there we go. This is a cross bridge right there. The myosin head and the active site of actin bind. This is a cross bridge. And now you could see there's another cross bridge here, how when the myosin head, which actually has a little hinge right there, when it pivots, it will drag with it the thin filament towards the H zone. And that's how the sarcomere will, on both sides this is of the H zone, this is happening, and the sarcomere is going to contract as it cross bridges are formed here and here, and the pivot of the myosin heads are going to bring thin filaments towards the middle. Okay. Now, the theory that explains how muscle contracts is called the sliding filament theory of muscle contraction. Long name. The theory explains how muscle contracts from the point of the nerve of the neuron, the nervous system cell, activating, stimulating the muscle cell, which is the first step in muscle contraction. And this is one difference between skeletal muscle and cardiac muscle. Skeletal muscle cannot move on its own. It needs the nervous system to stimulate it, and then it will move, it will contract. Cardiac muscle is independent of the nervous system. It can initiate its own contraction. And we'll see that next semester, 2402, when we look at the heart. But that's a crucial difference. So without the stimulation from the nervous system neuron, the skeletal muscle will do absolutely nothing. Okay, so it needs a stimulus. So the first step in muscle contraction is the stimulus. Then that stimulus needs to be able to release calcium ions inside the cell. That's the second step. It's called excitation, contraction, coupling. So how does that stimulus cause the release of calcium ions? Once the calcium ions are released, now the active sites of actin are available and cross bridges are formed, which cause the contraction. So those are the three steps of the sliding filament theory of muscle contraction: the stimulation, the coupling between the stimulation and release of calcium ions inside the muscle cell, and then calcium ions are going to initiate the formation of cross bridges, which are the actual contraction. Now, I do have some, right here, animations right here on this muscle contraction. There should also be animations of visible body too for muscle contraction. So it is helpful to view animations of these events to be able to understand them a little better. Having said that, let me go through some of these in a very summarized manner. So the very first event, the very first step are called the events at the neuromuscular junction. Okay, neuromuscular junction. That is the place where the nervous system neuron and the skeletal muscle cell come together. Okay, that's the neuromuscular junction. So what we have is a neuron, which is a cell of the nervous system. And the little body of the neuron is inside the spinal cord. but the axon of that neuron leaves the spinal cord and begins to travel, sometimes very long distance, all the way to the skeletal muscle cell that it is going to stimulate. So you can imagine how the neuron that is in charge of or the neurons that are in charge of stimulating the muscles of the toes are very long neurons. They're going to have very long axons because they have to go all the way from the end of the back where they're going to come out of the spinal cord and travel down the leg all the way to the toes. And that can be, half a meter in some people, even longer in others. So those are incredibly long neurons. So these neurons that serve skeletal muscle cells have special names, and we're going to need to know them because from now on we're going to call them by their names. The name of this neuron is a somatic motor neuron. So when you talk about a somatic motor neuron, you're being very specific. You're talking about the types of neurons that serve skeletal muscle cells. The neurons that serve a smooth muscle are not called somatic motor neurons. The neurons that innervate the heart are not called somatic motor neurons. Okay, so that somatic motor neuron is specific to these kinds of neurons. So definitely know what a somatic motor neuron is. If not for this chapter, for the next. Okay, so these neuron and the skeletal muscle cell come together at the neuromuscular junction. So here's a depiction of where they come together. Let me see if I can think. Actually, let me do it this way. Okay, so let me go back really quick. All right, the end of the neuron right here, and then at the very end, the neuron kind of branches. And at the very end of the branch, there's a little bulb, which this is what it represents right here, that bulb, which is what this picture represents. So that's the end bulb of the neuron right there. And inside that bulb, there are these little vesicles filled with a chemical, which is going to be called a neurotransmitter. This is the muscle cell, which means this is the sarcolemma, the cell membrane. And this is the place where the neuron and the skeletal muscle cell meet. That is the neuromuscular junction. Notice that the two cells do not touch. There is a gap in between. There's a space in between. This is called the synaptic cleft. The space is essentially tissue fluid. It fills that space. Notice that the other end, where the muscle cell is, the area that of the neuromuscular junction has these little protein receptors right here. Okay, these are ion channels, and the neurotransmitter that is going to be released by the neuron is going to travel. Once it's released, travels into this cleft and binds to the receptors in the muscle cell, skeletal muscle cell. When the neurotransmitter, which by the way is called acetylcholine, short is ACH, definitely know that. We're not going to, acetylcholine is going to be with us this semester to the end and next semester. So don't forget it. Acetylcholine is the name of the neurotransmitter released by somatic motor neurons at the neuromuscular junction. So acetylcholine, ACH for short, travels down the little synaptic cleft and binds to the acetylcholine receptors in the muscle cell. And that happens, these receptors open up and allow sodium inside the cell. And sodium is positively charged. The cell is negatively charged. And now all of a sudden, a tremendous influx of positive charges go inside the cell, which create a voltage change in the cell. The sodium ions continue to go in and are going to go down the T-cells. So, just a second. I have a close-up of all the, okay, there you go. So there's the little vesicles filled with acetylcholine. One thing that happens right before the acetylcholine is released is calcium outside the muscle cell. This will be blood calcium that is released into these area. And here's one reason why blood needs calcium at all times. Calcium goes inside the neuron, and that's what kicks out the neurotransmitter. So here's the neurotransmitter traveling down, bound to the receptor right there. When that happens, the receptor opens and allows ions inside that create a positive charged area all around where the cell membrane is. So this is the stimulus. This positive ions going in is the stimulus that has finally reached the skeletal muscle cell. All right. When that happens, these flood of positive charges go down the T-tubules, and as they do that, they begin to open these electric gates, which are the calcium gates that keep the calcium hidden inside the sarcoplasmic reticulum. When the positive charges open the electric gates, the calcium ions begin to get out inside the cytoplasm. The troponin and muscle contraction begins. This event in which the positive charges travel inside into the T-tubules, inside the muscle cell, and begin to open up the calcium channels, this is the excitation contraction coupling step. Okay, and then we have the calcium being released. Calcium is going to bind to troponin. Okay, binds to troponin, and then troponin moves, drags with it tropomyosin, and now the active sites of actin have been made available. They're released. Myosin heads and active sites of actin come together like two poles of a magnet. The minute they're available, boom, they're going to come together. Another important point, notice how the formation of the cross bridge does not need ATP. There's no ATP in that picture. You do see calcium ions, but you do not see ATP anywhere. Energy has not been used at all for any of these events. Okay, let's continue. Now we have a cross bridge. Now we are into the contraction part of the cycle. Okay, so all right, here's your cross bridge right there. Notice that the myosin head does have an ADP, which is a spent ATP. Remember, ATP is broken down into ADP and phosphate. So the spent ATP is attached to the myosin head. We haven't used any ATP yet. So these came from the previous contraction, or the previous cross bridge, I should say. Okay, so this is what the myosin head looks like when it makes the cross bridge. You can see the little calcium attached to the troponin. Look at what happens. ADP and phosphorus leave. The minute the myosin head binds to the actin, ADP and phosphorus leave. And you can see how when that happens. So look at the position of the myosin. Oh, there you go. There you can see it moving. Okay. Now ATP binds to the myosin head, and look what happens. The minute ATP binds to the myosin head, that energy that's going to be released when ATP is hydrolyzed is going to force that myosin away from the cross bridge. So now we have cross bridge detachment. The ATP is hydrolyzed, and now the myosin head is again an ADP phosphorus attached to it. Now it's ready to make another cross bridge, bind to the active site. ADP and PI leave, and the little myosin head moves. You can see here moves, all of them, not just one, but all of them move towards the middle, making the myosin move. Then ATP comes in, binds to it, and forces it to detach. The ATP is hydrolyzed, and now the myosin head is ready to start over again. And so these constant cross-bridge formation, pulling the thin filaments towards the middle, detachment of the cross-bridge because ATP energy has to be spent to detach. And then again, another cross-bridge, and then this continuous cycle is what causes this muscle contraction. Okay, as long as calcium is available and keeps the tropomyosin out of the way, and active cytobactin is available, this will continue to happen. So this is a continuous event as long as calcium is available. And calcium will be available for as long as the muscle is being stimulated. Okay, so this is a constant cycle. All right. My point here now. First of all, what do we need to know from here? Obviously, you need to know what a cross bridge is. Cross bridges are very important. Which proteins are involved in a cross bridge? In the actual cross bridge. In the actual cross bridge, you have actin and myosin coming together. They're the ones that make the actual cross bridge. ATP, the role of ATP is to detach the cross bridge. So you need energy to break the cross bridge. You do not need energy to make the cross bridge. You need it to detach it. Again, just like you do not need energy to bring the poles of two magnets together, but if you want to separate them, you're going to have to force them separated. That's exactly the same principle. All right, what else do you need? You need calcium to make the active cytobactin available. Okay, so have an idea of the role of ATP, an idea of the role of calcium in all these. I'll definitely know what a cross bridge is and which molecules are involved in a cross bridge. Okay, now this is just going over the whole process again, and it's just definitely know what changes in the sarcomere during muscle contraction. Now, from these, you should be able to determine what happens, for example, if, let me go back here, if, for example, all the way back, yeah, right here. Okay, what would happen if you introduce a toxin in this mechanism that prevents the release of acetylcholine, which is what Botox does? What would happen to muscle contraction? Would you expect muscle contraction or no muscle contraction? What happens if you eliminate calcium from the picture? Would you have muscle contraction? Okay. What happens if when a person dies and there's no more ATP available? Would muscle contraction be stopped at the cross bridge right here, or at the relaxation step right here? You need ATP to detach the cross bridge. If there is no ATP because the cell has died, this is where you're stuck. So that explains why after a person dies, the body becomes rigid. Rigor mortis sets in, because this is where muscle contraction stopped, at the cross bridge. What requires energy is the relaxation step right there. Another step that requires energy is getting rid of the calcium. Getting rid of the calcium and putting it back into storage, that requires energy. So bringing all this calcium, this is called recapturing of calcium, bringing all this calcium back into storage, you need ATP for that. That's going to be to bring something against the concentration gradient. So forcing the calcium back in, that will require energy. So again, after death, there is no energy to do that. Calcium is permanently out in the cells, and so therefore you're going to end up with muscle contractions. So so you have to have an understanding of it enough to answer questions about it. You're not going to be asked to explain every one of these steps, but to have somewhat of an understanding to be able to answer questions about the steps. Hopefully that makes sense. One more thing I did not mention, let me just find it real quick. Another term you need to know, definitely know acetylcholine as the neurotransmitter. Now, acetylcholine does not stay around for very long. It is removed very quickly. And that is in part due so that we can control muscle contraction better. There's an enzyme called acetylcholinesterase, ACHase for short, which is always found in the synaptic cleft. So acetylcholinesterase is always here, and the minute acetylcholine is released, it begins to break it down, so that the acetylcholine is not going to just hang around and continue to stimulate the muscle as we want to. And as we continue to send signals, acetylcholine will be removed very quickly. And that gives us more control over muscle contraction. All right, any questions? This is the hard PowerPoint right here, the first one. The next ones are not so bad. I have a half an hour. What I'm going to do is I will go over and tell you what to know or what's important. Definitely know what a motor unit is, which is the organization between neurons and skeletal muscle cells. And notice that one neuron is actually in charge of more than one cell. Okay, so that little red neuron is in charge of 3 cells. That's a motor unit. One neuron and the cells that neuron is in charge of. Motor units work asynchronously. Obviously, in one muscle, you're going to have many motor units, and they don't work all together at the same time. Otherwise, the entire muscle will tire very quickly. They take turns. And when one set of motor units are contracting and begins to relax, the next set of motor units takes over. And that allows for being able to use the muscle for a longer period of time. Definitely know what a twitch is. Expect to be able to label the parts of a twitch or to understand which ones are the parts of a twitch. A twitch is one stimulus given to a muscle, not a muscle cell, but the entire muscle, so that you can see the electrical pattern of muscle stimulation, muscle contraction, muscle relaxation. That's what a twitch is. One stimulus into one muscle that gives us the contraction and relaxation period. First, we have what we call the latent period. This is the excitation, contraction, coupling period. So the stimulus is the first part of muscle contraction. These are the events at the neuromuscular junction. This is the first part of the filament theory of muscle, sliding filament theory of muscle contraction. Then the lag period, when nothing is happening yet, is when the stimulus goes inside the cell and releases the calcium. And then we can see the cross bridges when we see the period of contraction. Okay. So we have the stimulus, the lag, or latent period, the latent period. Then we have the actual contraction that increases with time. Notice how short the time is, however, as more and more cross bridges are made. And then the stimulus, the acetylcholine has been removed, calcium begins to be removed from the system, and the muscle relaxes as calcium removed. That's one twitch. Each muscle has different types of twitches. In general, muscles over which we have fine control are going to have short twitches, small twitches, if you will, and small motor units, meaning that one neuron will be in charge of only 1 or 2 little cells. These are the muscles that we have a lot of control over. The muscles that move the eyeball, the extraocular eye muscles, The muscles of the fingers, especially the thumb, very fine control over those muscles. We can write, we can paint, we can do all kinds of intricate work with those muscles. Those muscles have short twitches and small motor units. So you need many neurons to serve those muscles. Muscles over which we don't have fine control have longer twitches and large motor units. Okay, so know what a twitch is. Know what we mean by incomplete tetany. Incomplete tetany is what happens when we stimulate a muscle repeatedly over a short period of time. So the muscle contracts and begins to relax, but before it has completely relaxed, we stimulate again. So now more calcium is released into the system, creating the next stimulus will create a stronger contraction. And before that muscle relaxes, we stimulate yet again. Not all the calcium has been captured by relaxation before we release more calcium, so the next contraction is even stronger because the more calcium, the more short cross bridges. And we'll do this repeatedly until we get maximum contraction. So when you're trying to lift something heavy and it's not working for you, and you stimulate even with a stronger contraction, and then stronger and stronger and stronger until you get maximum contraction of so that you can lift that heavy thing, this is what you're doing. You're going these incomplete tetany events. Now, once you lift something heavy above your head and you want to keep it there, a heavy weight, this is what you're going to be doing. You're going to stimulate your muscle in such a rapid frequency that there is no relaxation. The muscle very quickly goes through maximum contraction, and the maximum contraction is sustained over the period of time, as long as these stimulus keep coming until relaxation comes. This is complete tetany. These are typical characteristics of a skeletal muscle. Smooth muscle and cardiac muscle do not behave in this way in response to stimuli. Okay, the next PowerPoints make a lot of sense if you read them. The more you stimulate, the more motor units you're gonna call into action. You should know what muscle tone is, slightest stimulation, all the muscles exhibit at any time. Know the difference between isotonic and isometric contractions. This is isotonic, this is isometric. Examples: isotonic is when you lift something. Isometric is when you carry something without lifting it, like a grocery bag. Muscle metabolism. This one is important too. Okay, this is what you do here. Skeletal muscles are specialized to make energy, to make lots of ATP, because they need lots of ATP. So they have a specialized way of making ATP. There are three ways. One way of making ATP is unique to skeletal muscle, and it's called direct phosphorylation. Skeletal muscle has a molecule called creatine, which can be phosphorylated. A little phosphate molecule can be added to it. And then, thanks to a special enzyme called creatine phosphate, CPK, the phosphate is removed and given to ADP. So ADP, creatine phosphate is going to remove that phosphorus and give it to ADP, so that at the end of the reaction we'll have creatine without phosphorus, and now we're going to have ATP because that phosphorus was given to ADP. And this is a one-step mechanism to make ATP. Very, very quick. So that's unique to skeletal muscle. It's called direct phosphorylation, and you can see it here. So this makes enough energy to keep us going for about 15 seconds, which is not very long, but still. Okay, so what if we need to create lots of muscle contraction over a short period of time. So if we contract the muscles forcefully over a short period of time, that is going to bulge the muscle. And when you bulge the muscle, that compresses on blood vessels and that impedes oxygen delivery to the muscle cells. So now the muscle cells don't have the oxygen to make lots of ATP. So they have to switch to an anaerobic pathway, which is fermentation. So now the muscles begin to ferment in order to make ATP. And actually fermentation allows us to make ATP from glycolysis. Fermentation is what keeps glycolysis going in the absence of oxygen. Problem is, we're only making 2 ATPs per glucose molecule. We're accumulating lactic acids. So that means that we're accumulating waste, making very little energy. We're not going to be able to do this for very long. So sustained strong muscle contractions is going to force us into an anaerobic pathway in which our muscles are going to run out of energy very quickly and accumulate waste. So we're not going to be able to sustain that kind of strenuous activity for very long. But the good thing is that we can sustain it for a little while in the absence of oxygen, which is a tremendous advantage compared to other cells that cannot do that. Now, when everything is good and we have lots of oxygen, now we can make tons of ATP. We have muscle cells, the skeletal muscle cells have stored glucose in the form of glycogen that no other cell outside of the liver has. It has stored oxygen in the form of myoglobin, which makes the muscle red, which allows it to make tons of ATP as long as oxygen is available. And this is what sustains long-term work. 32 ATPs per glucose molecule versus 2 with glycolysis. So this is what we do when we're engaging on everyday activities, aerobic exercise, anything that is mild activity that allows for oxygen. The minute we have very strenuous activity with tremendous muscle contraction that is going to impede the flow of oxygen, now we're going to run out of steam very quickly. So it's okay for short duration exercises, weightlifting, sprints, but you cannot sustain it for very long. Sustained reactions are the ones that allow muscle to make ATP with oxygen through aerobic respiration, and that's going to keep us going for all day long. Marathon runner. So I will let you read all these. I do want to go for another part really quick. So I have an idea of how muscle makes its energy and be able to recognize the 3 ways in which the muscle makes its energy. Now, out of these, PowerPoint C, this is what I want to go to. We have a few more minutes. Okay. All right, this talks about the different kinds of muscle cells that we find in muscles. So not all the muscle cells within a muscle are going to be the same, especially in humans. We have a mixture of cells. Some cells are slow oxidative fibers, all others are fast glycolytic fibers, others are something in between, fast oxidative fibers. We're not going to concentrate on the middle ground cells, we're going to concentrate on the extremes, slow oxidative and fast glycolytic. Those are the ones we're going to concentrate on. The middle ground is something between those two. So you have to know the characteristics of the slow oxidative fibers and fast glycolytic fibers. So at this point, you have 2 choices. You can memorize the characteristics of each, these and these, or you can use what you already know to very quickly come up with the characteristics of each. So this is a slow oxidative fiber. This is a fast glycolytic fiber. When you look at this picture, the most striking thing you see when you look at the picture is the difference in color between the 2 of them. One is light, the other one is dark. Okay, the color is due to myoglobin. So this guy, the slow oxidative, has myoglobin, which is a molecule that stores oxygen. which means that cell is going to have a lot of oxygen in it, which means it's going to allow it to uh, to um, to work for a long time. So it's fatigue resistant, doesn't fatigue easily. So it can go longer. This one, on the other hand, is going to fatigue easily. Okay. Second thing is the fast glycolytic is bigger than the slow oxidative. If it's bigger, it means it has more myofibrils. If it has more myofibrils, that means that it can give us a stronger contraction. So it can give us a stronger contraction, but it's going to fatigue more easily because it doesn't have the oxygen for sustained muscle activity. It's going to have to switch to fermentation very quickly and get tired. Versus the slow oxidative has oxygen storage, which will allow it to go on for a longer time, but it's not going to give us as strong a contraction because it doesn't have as many myofibrils. Let's see. We got dark color. We got that one. Okay. Yeah. From there, you can extrapolate. The slow oxidative is using oxygen, which means it could also use fatty fats and proteins, not just carbohydrates, to make energy, versus the fast glycolytic, which uses glycolysis only to make ATP, which means it is reduced to using carbohydrates only. It cannot use fatty acids, it cannot use proteins. So it has to have a reserve of glycogen. So it's going to have more glycogen reserves, so more glycogen. The slow oxidative will have more mitochondria because mitochondria is where ATP is made with oxygen. It's going to have more blood vessels because, again, it's going to it's going to need more oxygen. Okay, so those are the basic differences between those two kinds of cells. So, for example, in a marathon runner, because, let me go back, the middle cells could be changed through training into one of the extremes. So a marathon runner would change its middle cells into slow oxidative cells. So it would have more slow oxidative cells than fast glycolytic cells. A weightlifter would have more fast glycolytic cells than slow oxidative cells. Next time, if you eat chicken, notice the difference between the dark meat and the white meat. The breast is the white meat. That's your fast glycolytic fibers. That is the breast of the chicken, which moves the wings. Chickens don't fly all day. Chickens only fly once in a while when they're trying to fly away from a predator. So they need a strong, big muscle to quickly make them fly away from a predator. A chicken stands on its legs all day walking. So that's where the slow oxidative fibers are, on the thigh. Same principle, more dramatic than in a human, but it's the same principle. So definitely know the difference between the slow oxidative and fast glycolytic fibers. Okay. And then the last part of the PowerPoint goes back to types of muscle tissue. So go back and review the characteristics of a smooth muscle, cardiac muscle, and a skeletal muscle. Which one has striations, which one doesn't. If it has striations, that means it has sarcomeres, which, how many nuclei on each, how many what else? The size of the cell, which one is involuntary, which one is voluntary. One thing about a smooth muscle, which does not have sarcomeres, it has no striations. That means it's not going to have the troponin to bind calcium. Instead, it has another protein called calmodulin to bind calcium. It's in the PowerPoints. Let's see here. So we'll put somewhere in here is calmodulin. So notice how smooth muscle contracts. It doesn't contract in this manner. it contracts all over because it doesn't have sarcomeres. Somewhere in there. Okay, somewhere in there is calmodulin, which is the molecule that binds calcium. Know the difference between, give me a second, think hyperplasia and hypertrophy. I'm sorry, I'm just kind of jumping around because I have another class actually. Okay, hyperplasia. Hyperplasia is what the smooth muscle exhibits, which means it's the when we increase activity of the muscle begins to divide and we get more muscle cells. Cardiac muscle and skeletal muscle, they don't exhibit hyperplasia, they exhibit hypertrophy. And I know this is in the PowerPoint, I'm going backwards because I know it's in the class. Yeah, so skeletal muscle exhibits hypertrophy. When we increase the activity, when we stimulate the muscle more, the response of the muscle is not to make more cells, is to make bigger cells. So the cells that it has become bigger. So the muscle grows bigger. And then sarcopenia, at another point in the PowerPoints, they'll talk about sarcopenia, which happens with age. Muscle cells become smaller. Okay, so know those terms. All right, questions? Okay, so if there are no questions, I'll go ahead and stop the